Good evening, everyone. So thank you so much for being here to attend to this session. And then, I know this is a Saturday, but everybody still make time to attend this live session about whole genome sequencing for PGTA, which is a very interesting topic. It's something that I always wanted to know more. So I'm very happy to have Dr. Shah to be here tonight to give us a talk about the whole genome sequencing application and to clinical embryology, PGTA, PGTM. So I'm going to just give a very brief introduction of our organization, International IVF Alliance. And we have some members who contributed to today's session. So I am your host, Ife Lu, and also three people who helped a lot with the preparation of this talk, who is Wei Zhang, and she is the lab manager at Reproductive Partners Fertility Center, San Diego, and Dr. Yu. Dr. Yu is the director of reproductive clinical science, PhD and MS program. She's also associate professor at EVMS. And also Yilin Li, she is the senior embryologist at Sutter Health, San Jose. So also want to introduce about Dr. Shah. He got his bachelor's degree at the University of Minnesota and also became a PhD student of UPenn and did his postdoc at Stanford University. And since 2022, he joined the Orchid Health and he's been the principal scientist. And he's the one who is leading the development, validation, and launch of lab-developed PGT testing for whole genome sequencing screening. So I believe today's talk is going to be extremely exciting. I've been looking forward to it for a long time. So Dr. Shah, please take it over. Thank you so much.
All right, thank you. Let me share my screen first.
Okay, let me stop sharing.
Can you see my screen?
Yes.
Okay, so first I would like to thank the organizer for the very kind introduction and also giving me the opportunity to share our study here. So my name is Yun Tao. So today I was told it's going to be a lecture series. So the focus will be pre-implantation genetic testing. And at the same time, I bring something new here. It's called pre-implantation genetic testing, whole genome sequencing. So not only just the PGT, but I also talk about the new assay, performance, validation, case study, and also talk about the assay and future of the field. This is our agenda for today. I'm going to talk about the current stage of PGT first, and also whole genome sequencing and why it is difficult to use it on embryos, and also want to go through some of the clinical validation, share some case studies, and also talk about ethics and future. So PGT has been clinically available for decades. So nowadays, the IVF centers can take biopsies from day 5 to day 7 embryos without hurting too much of embryos viability. So, and then the PGT lab will take the biopsies and do genetic screening on those. There are three major categories of PGT. The first one is called a PGTA. It is a very simple assay, mainly just counting chromosomes. For example, trisomy 21, trisomy 13, trisomy 18, etc. The second category is called a PGT-SR. It is for chromosome structure rearrangement. Mainly, most people use it have translocations. It is actually, in practice, it is actually just a subset of PGTA. So basically, in PGTA, people look at the whole chromosome gain and losses, and large chromosome segment gain and losses. For PGT-SR, because some of the translocation are smaller, so that in practice, the lab just sequence the PGTA sample a little bit deeper so that they can get a little bit higher resolution on the chromosome segments. So overall, it's not that exciting because it's just a PGTA, a deeper sequence version of PGTA. And many labs do not even distinguish the balanced translocation carrier versus normal euploid embryo, so that you can see there's still a lot of room for improvement. The PGTM is another category, so it screens for single gene disorders, for example, BRCA1, CFTR for cystic fibrosis, etc. There, it sounds very promising, but it is actually very, very limited. For example, there are hundreds of pathogenic locations on BRCA1. However, if you want to use it, the most you can do is only screen for one or two positions only. And you need to go through a diagnosis first or screening first to make sure you're either affected or a carrier status. Otherwise, the case will get rejected. If a couple want to say, okay, I just want to screen my BRCA1, then that's not how the PGTM is working. So basically, you can see overall PGT is still very, very limited. And there's still a lot of patients get rejected by PGT company every single day due to the test limitations. For example, we got the patient like as a translocation, one at the chromosome 17, 2 megabase pair, another one at the chromosome 22, 4 megabase pair. So the patient went to Natera first. Natera said, okay, our lower limit of detection is 7 megabase pair. I cannot do anything for you. So they also get rejected by other labs, and at the end they came to us. Same thing happened to other patient. We got a patient with a de novo variant in their first kit, causing neurodevelopmental disorders. So after that, all they want is, they want to screen for the embryos to make sure the same pathogenic variant does not happen in the other embryos. And they get rejected by other PGT labs because they don't deal with the de novo variants like this. Also, their situations, the patient with like micro deletions, micro duplications that are like 400 kilobase per 500 kilobase per size is very difficult to identify, so they get rejected. And PGTM cases as well. The parents are not in the United States. The parents passed away. The first degree relatives do not want to provide their samples. This situation happens every single day. And so we're asking, is there a better option that can not only cover the current PGT, but also actually fill the gap and accommodate the people in need? We're thinking about the whole genome sequencing, and whole genome sequencing becomes more popular these days after the reagent costs were significantly reduced. You can see there are many papers published in Nature Science, all these high impact journals. They are using whole genome sequencing on rare diseases, very complicated genetic conditions. The reason for that is the whole genome sequencing now not only look at the SNPs or indels, but also look at complicated genetic conditions like microduplication, microdeletion, uniparental isodisomy, etc. And this is actually much more thorough than the traditional targeted sequencing, and sometimes it's faster because sometimes people go through the genetic testing. In the United States, they try panel A, nothing positive, and then they try panel B and nothing positive, and they try panel C. So this is actually very time consuming and energy consuming process as well. So whole genome sequencing give people lots of data, very thorough, so people can analyze many genes and many different conditions at the same time to find the cause. However, applying whole genome sequencing to embryo is very difficult, mainly due to the DNA in biopsy is very limited. So basically, if you want to do a medical grade whole genome sequencing, you need at least 100 to 500 nanogram of DNA. However, in a trifecta biopsy, there's only 30 to 50 picogram of DNA available. So you can see, one is picogram, one is nanogram. There's still a very big gap, and you need a whole genome amplification in order to get enough DNA. But many of the whole genome amplification methods are not ideal. So before I go into the detail about the whole genome amplification, so how the PGT is done. So based on my flowchart, you can see the workflow is actually very straightforward. The lab receives the biopsy first, and then we add the lysis buffer in the PCR tube. The lysis buffer will open up the cell membrane and also open up the nuclear membrane as well, so that DNA will be released in the tube. So you can see there's no DNA extraction. Everything happens in the same PCR tube you collect the biopsy. And after the DNA is released to the lysis solution, we can perform a whole genome amplification. Um, Depends on the purpose of the assay. It could be PGT-A, PGT-M, or other assays. You can choose different whole genome amplification method. I'll talk about those in later slides. And after a whole genome amplification, you can get to enough DNA for your downstream assay. You can either load the DNA into the array or load the DNA into the next generation sequencing. And after that, you do data analysis and you can report the result to the IVF centers. So let's take a look at the result from NGS versus the result from the array. So this is kind of like a result from the NGS. All this plot is all you can get from NGS, low pass sequencing. For PGT-A purposes, only low pass is performed. The people focus on higher and lower bins. Those higher signal indicate a gain of the chromosome, and those lower signal indicate a loss of the signal. And sometimes when the resolution is higher, you can see the segmental gain and losses of chromosome as well. Here's a result from the array. You can see the array data is more informative. You not only have this binning counting, but also have this B audio frequency information. For example, if you see a lower signal, is it an artifact or is it a real loss of chromosome pieces? And then now you can look at the beta audio frequency. You see a loss of heterozygosity here and of those SNPs. So now you are very confident. Okay, this is a true signal. This is telling me, the lower signal plus the loss of heterozygosity of the beta audio frequency. Now I'm very confident this is a loss of the chromosome. And also here you see a bump here. So is this elevated signal again of chromosome? The answer is yes, because when you look at the beta audio frequency, you see this very clear audio splitting. This audio splitting is a kind of like evidence to show you, okay, there's a gain of chromosome there. So you can see in NGS data, you only have this binning approach and counting. In array, you actually get a little bit more information. You get the beta audio frequency to confirm your result. Overall, you can see when you work with PGT companies, array is $50 more expensive than NGS. However, the data I showed you here, just for an example, both of them are from the genomic DNA or from blood or cell line, those genomic DNAs. The data looks actually very clean, and that's why you can do the analysis like this. In embryo, the data looks a little bit messier because there's a whole genome amplification in between that kind of lower the quality of the DNA. So this is overall a PGT company's PGT-A, and you can see definitely the dots are more scattered than the genomic DNA that I just showed you. However, you can still see there's a trisomy 21 here. And this is the array data. The array, you see an elevated signal here. You kind of see the audio splitting. And this is, this kind of data like processed by PGT company, every single day. This is our data, whole genome sequencing of the embryo. And you can see the dots are smaller and are much more tighter. And in ORCID, we do whole genome sequencing, and the industry standard for whole genome sequencing is 30x steps. So we sequence the sample to 30x steps because it's very deep sequencing. We also get beta audio frequency. We're not doing array, but because the sequencing level is very deep, we also get this audio frequency. So basically, you can see this sample has a trisomy 21 and also a monosomy 22. So for monosomy 22, as I mentioned, usually is supported by a loss of heterozygosity, and that's exactly what I see here. So basically, this give me very high confidence that this sample has a loss of chromosome 22. And for chromosome 21, we not only see the gain of the signal here, but also we see this very clear audio splitting and that gives me very high confidence that there is a trisomy 21 here. So so in ORCID, although we use NGS, but we have orthogonal data to confirm, to add our confidence about our calling. And why sometimes the dots are very scattered, sometimes the dots are very tight, is all due to the whole genome amplification method. There are different whole genome amplification methods on the market. For example, the first one is called the DOP PCR. It is a two-step PCR process. The first one is called a low temperature PCR. Why is called a low temperature PCR? Because the binding temperature during this PCR is very low. So basically, also you can see the primer is all randomers on the 3'. So there's no specific primer. They're all just random sequences. Because the binding temperature is low and the primers are all randomers, basically the primer can bind wherever it wants. And after this low pass PCR, this genome get probed all over the place, from chromosome 1 all the way to chromosome X, all over the place, get primed. And after that, the yield is actually quite low, so you want to do a PCR to enrich the PCR product from your first amplification. And then you take advantage of the blue hinge and do a PCR for enrichment. And after that, you will get enough DNA for the downstream assay. So the advantage of DOP PCR is usually the uniformity is very, very good. So basically, it's ideal for counting chromosomes. So some of you may be lab directors or doctors or embryologists. You see PGTA report, right? So if you see the method section, it talk about DOP PCR or picoplex or shortplex or anything related to PCR. The core is the DOP PCR. Although shortplex, picoplex, those are like upgraded version of that, but the core, the chemistry is the same. So PTA usually the DOP PCR. The second method is called MDA, multi-displacement amplification. It is called a multi-displacement amplification because the DNA polymerase can displace the previous strain, double-stranded DNA, can displace the previous strain. So the DNA after get displaced, they becomes like hanging tail in the solution. But this tail can be very long. It can be like 10,000 base pair and even higher. So that becomes a new template for more primers and DNA polymerases to bind for further DNA amplification. The good thing about this method is the genomic coverage is very, very good. So usually when you need to do PTM, you want to cover your region of interest during whole genome amplification, and MDA is commonly selected for these purposes. The problem for this method is the uniformity. And you can see because the formation of this tail, you can see after a few run of amplification, some region get over amplified, whereas some region get under amplified. And overall, when you sequence the sample, you can see they're not very uniform. You see spikes. Those spikes are like over amplified regions, and those little blue bars are like under amplified region. So you don't see a uniform line here. You see spikes. So this is also not good. So in 2021, when I was a postdoc at Stanford University, I was involved in developing a new whole genome amplification method called PTA. So what PTA is a modified version of the MDA. What it does was, it used a special molecule called irreversible terminators, so that we can block the amplification at a very short length. So basically, amplify, stop, come off, amplify, stop, come off. That's the mechanism of this amplification. So basically, because we stop the amplification at a fairly short length, so we can prevent the formation of this very long tail. And as a result, we can prevent the formation of this very bulky, ununiform structure. And when you sequence the sample, you see this blue line is much more uniform compared to those blue spikes in the MDA. We published the work in 2021 in the PNAS, and my last project at Stanford was to optimize the system and try to apply it in the human embryo. I'm the first one who actually did it. So the next question is, are any of these methods good enough for whole genome use? So let's revisit, what I just said. So for DOP PCR, it is a very uniform method. It is ideal for PGTA. So either picoplex or shortplex, those are upgrade version of DOP PCR. However, the problem for this method is, regardless how hard you sequence, the maximum genomic coverage you can get is 30 to 40%. So for whole genome sequencing, we really want to cover about 100% of the genome. 30% and 40% of genome is not ideal in these purposes. So how about MDA? MDA has much greater coverage. However, as I said, the uniformity is very poor. Some is over amplified, some region are under amplified. So when you sequence a standard 30x whole genome sequencing, only 60% of the sample are above region are above 10x. So this is not ideal. So we lose a lot of regions. So we cannot use that. And you can see MDA has a lot of scatters, not very uniform, consistent with what I just mentioned. But it can still be used for PT a little bit, because this is the trisomy 9 sample. You can still see the trisomy 9. That's why when people do PTA and the PTM at the same time, they can still use MDA to do this type of assay a little bit, whole genome amplification. Not a huge trouble, but just not ideal. How about PTA? I mentioned PTA is actually improved from MDA. Is it better? So usually the DNA genome, like human genome, right, is consist of intron regions and exon regions. Exon regions is where all the genes are located, and they are only 1% of the genome. We want to make sure not only just overall cover the genome, but also those selected 1% need to be well covered. This is much higher requirement than just you get the normal uniform whole genome amplification. So when we look at the PTA, unfortunately, PTA missed 25% of the exon regions, making it less ideal for whole genome use. Later on, we realized this irreversible terminator is actually very harsh to the enzyme, also the reaction. So that we still follow the same logic. We believe the logic is correct. We want to shorten the length of the DNA during amplification. We switch to other method to shorten the length of DNA during amplification. And this is the first time we see that we get 96% of the exon coverage, and also 98% of exonic sensitivity, and without introducing much of the false positive results. And this is the first time we say, okay, PGT whole genome sequencing could be feasible. So after that, we started our validation. We want to validate, whether we can actually use it in the clinical setup. We first validate our method against the genome in the bottle cell line. Why we choose genome in the bottle cell line? Because the entire genome was fully characterized by the National Institute of Standard and Technology. So all the sequence, the entire genome sequence, get published online, so we can use. And also because the entire genome is published, it's easier for us to calibrate whether we can our method has systematic false positive or systematic false negatives. This is how we did it. We diluted our DNA to 50 picogram. This is amount of DNA I mentioned. The trifectodon, available in a trifectodon biopsy. So we use the DNA from the same level. We perform our whole genome amplification, followed by a whole genome sequencing. And after that, you can see the result from the table. We get very decent accuracy, specificity, 98% of precision, and 98% of the sensitivity. This is excellent. So compared to the golden standard genomic DNA, is almost comparable to the data coming from the golden standard genomic DNA. But remember, we get to this result from only picogram of DNA. So next, we're very excited about the results. So next, let's we say, okay, let's validate our assay using the real sample type, the biopsy and the embryo. We use the embryo as the reference, and then sequence our biopsy and compare the concordance between the biopsy and the embryo. And again, the concordance level is very high. This 98% of the sensitivity and 98% of the precision with very decent accuracy and specificity. So basically, the result tells me that when we do whole PGT, whole genome sequencing on the biopsy, the data we got can reflect the whole genomic information of the embryo. We also validate against the PTM samples. Those samples, those embryos, were donated for research, so the patient doesn't want those anymore. Those were tested by the third-party PTM company before. Um, so basically, for the regular PTM, those embryos were design probes, need the parental DNA, spend all the effort to just probe one or two locations accurately, for the variant of request. For us, it is expected that the whole genome sequencing should be able to detect the variant on the entire genome and should be able to capture this location without any problem, because theoretically we sequence every single position on the genome. And in this study, we did not collect the parental data. We do not need any probe design. And then just the raw read out of the sequencer, we got consistent result on both positives and the negative samples. So we say, okay, this is excellent. We read the entire genome, and our accuracy is as good as the PGTM assays. And also, we validated more complicated genetic conditions, for example, triploid, uniparental isodisomy, and some specific microduplication and deletions. For example, the crater cat, D. George, Prader-Willi, those are like higher prevalence microduplication, deletions, or UPD situations, and we were able to detect that all those without any problem. So just give you some example how the data looks. So basically, there are two samples, two potentially euploid samples under regular PGTA through NGS. Both of the sample will be considered euploid female. However, if you look at our data, because we have the beta allele frequency, only the first one is a true euploid. The second one is actually a triploid female. So basically, if you transfer the second embryo, still it can get to pregnancy, but the fetus will stop growing after 2 weeks. And this is a DeGeorge sample. So on the regular PGTA, it will be a euploid male. However, in our assay, we see a 3 million base pair deletion on chromosome 22. So if the baby was born, then the baby will have a neurodevelopmental delay, heart disease, as well as other phenotypes. This is another sample with Williams syndrome. So basically, on the regular PGTA, it will be euploid male. However, under our assay, we can tell that there's a 1.4 million base pair deletion on chromosome 7 that causes the Williams syndrome. And this is the UPD case. So for again, if this is just a regular PGTA through NGS, then it's gonna be a euploid female. But actually, the embryo has a UPD on chromosome 15, causing the Prader-Willi syndrome. So just summarize the first part. So we validated our assay with the genome in the bottle cell lines, embryos, biopsies with very high accuracy, specificity, sensitivity, and precision. We also performed a confirmatory test with the previous detected SNPs from donated PGTM embryos, and also triploid UPD microduplication deletions, those more complicated conditions, to make sure our assay is compatible for very complicated analysis. So we published our PGT WGS validation in FF's report in early 2024. However, our research never stops there. We just got an IRB approval to study the concordance between PGT WGS versus the resulting baby. And we just start to collect the pregnancy data and also the live birth data. I don't have the pregnancy and live birth data right now, but maybe later in ASRM or other conference when we met, we can talk about it. But I just don't have those right now. But I can talk about the concordance analysis at this stage. So this is the first case we got, for this study. There's a couple. The couple went through the IVF cycle, had three embryos. They transferred the first euploid embryo. It was a success pregnancy, then live birth of the baby. During the delivery, the cord blood was collected, followed by a whole genome sequencing. And our goal is to compare the concordance between PGT whole genome sequencing and the cord blood whole genome sequencing. This is actually a very interesting study because the biopsy for this embryo is actually at a lower quality site. It passed the QC, but very marginally. So we think it represents a good test of the technology to demonstrate a lower bound performance. And even at this quality, you can see we still get to 96% of the panel coverage. And I'll talk about this panel in the later slide, but you can see, it's still a very decent data. Uh, on the panel, we see 1422 variants in total in the cord blood, and we use it as a reference. In embryo, we identified 1385 true positives, missed 37 variants, and 32 false positives, accumulating a sensitivity of 97.7% and also a precision of 97.4%. So you can see the number agrees with what we promised in our validation as well. So among these 1400 variants, how many of them are clinically relevant? Not many. The mother has two, the father has one, the cord blood has two, embryo has the two and the same two. So basically, for inherited variant, cord blood and the embryo already are consistent. And how about the de novo variant? So we do not see any pathogenic de novo variant in the cord blood. We also do not see any pathogenic de novo variant in the embryo. But we still want to do a proof of concept study to see if there are de novo variant in the baby. Can we see that in the PGT WGS data? So we did analysis. We identified 16 possible de novo benign variants in the baby in the cord blood. And we only have limited amount of DNA, so we randomly selected 5 for a Sanger confirmation. And the Sanger confirmed that all 5 variants are de novo. They're not in father, not in mother, only in the baby cord blood. And when we go back to the PGT WGS data, all 5 variants get captured accurately in the PGT WGS data. So again, these 5 de novo variants were not reported because they were classified as benign, not because we did not capture them. So overall, although we understand we need a more data size and sample size, and we are working on it, but so far the preliminary data already show that the high concordance level between the cord blood, the baby, and also the PGT whole genome sequencing. And it doesn't matter it's inherited variant or de novo variant, it looks like we can capture both. So in terms of a product, what is actually offered to patient in the PGT whole genome sequencing? We're definitely not doing the whole 20,000 genes on the human genome. We actually, some genes are more clinically relevant than the others. So basically, we curated panels for reporting. So basically, we do whole genome sequencing to collect all the data, but for reporting, we do selected reporting based on panels. We curated genes for neurodevelopmental disorders, cancer panel, birth defect panel, and the ACMG secondary findings. So overall, altogether, about 1,200 to 1,300 genes in total. Especially the ACMG secondary findings, most of them are autosomal dominant, and you don't see them in most of the carrier screening report. So besides that, we also focus on complicated genetic conditions like triploid, UPD, microduplication, microdeletions. One of the common question we get from the doctors is, so how do you know, if you find a small deletion, small insertion, how do you know they're pathogenic, right? So that's why for microduplication and microdeletions, we do targeted analysis. So we have a list of 50 regions that are already proven to be clinically relevant and pathogenic. Those include like DiGeorge regions, Prader-Willi region, Prader-Cat region, Williams syndrome region. So as long as we see those in the embryo, we know the embryo is affected. There are also a PGTN category here, but overall, this is for like customer special request. So for example, the customer can have a variant in a gene that is not on our gene list. Can we accommodate? Yes. And sometimes the patient has translocation. The patient want like, okay, can you tell me whether it's whether my euploid embryo is a normal euploid or it's a balanced translocation carrier? Conditionally, yes. And so the patient can say, okay, I have a microduplication 400 kilobase per. Can you do that? And we will do that for sure. So although we call it a PTM, but it's actually a special request a channel for very complicated genetic conditions, and we do not charge any extra money for that special request. So let's look at some patient cases. So this is the first. So this patient actually wants to transfer as soon as possible. The female patient carries a HOG1 variant on her carrier screening report. The male patient carries another HOG1 variant on the carrier screening report. Usually, this is a PGT-M case. They need to talk to PGT companies, design probes, provide their DNA, and because there are two variants evolved in this case, so probably need to take a little bit longer time to design probes. But they want to transfer as soon as possible, so they came to us. We look at their carrier screening report. We accepted the case, so we did not need a parental DNA. There's no probe evolved. After 21 days, we returned the result. They were able to transfer. We had a more extreme cases. So that case actually is a male patient and a donor with a donor egg. The donor, when she donated the egg, she did not have the carrier screening report, and later on she did it. And then before the male patient wants to transfer and saw the carrier screening report of the female has a X-linked variant, as well as two other variants on the carrier screening report. And very unfortunately, the two extra variant match exactly with the guy had on his carrier screening report. So he requested a 3 variant, and at that time point, it's difficult to collect the female sample. So they get rejected by many PGT companies, and then they came to us. We can do that case without any problem. We don't really need the donor's DNA. So this is the case with a microduplication. The female was normal. The male partner has a de novo 400 kilobase per microduplication that causes a split hand foot malfunction. They don't want their kids to have the same thing. So they get rejected by other PT labs for multiple reasons. First is actually a de novo. Second one is it's a microduplication. It's very small. It's hard to see. So they get rejected. So we took the case. So basically, the father also gave us his sample for confirmatory purposes. That's why I make a plot here. So basically, you can see even though the pieces is very small, only 400 kilobase per, we can see that without any problem. You can see the elevated signal on the chromosome counting in the NGS data, and it is further supported by allele splitting. If you remember me talk about this allele splitting for trisomy regions. There are two alleles. The blue allele is the disease allele that has the extra copy. The red allele is the good allele from the father that does not have the extra copy. And you can see they're actually very lucky. All their embryo inherited the good allele from the father. And you can not only see the two the chromosome count pieces, the two copies from the NGS count, but also we have supporting data from the beta allele frequency to confirm the result. So we are very confident, even though the piece is very small, 400 kilobase per. We reported all this thing to the couple. The couple was very excited. I think part of the reason is all their 3 embryos are normal. this is another case. The patient was concerned about her family history of diabetes. She literally said that, there are crazy family history of diabetes in her family, but actually no one did a genetic diagnosis. She actually did a carrier screening report, but there's no finding at all related to diabetes. We performed the whole genome sequencing under embryos, and we found a GCK variant in about half of the embryos related to maturity onset diabetes of the young, called MODY. Subsequent diagnostic testing confirmed that the mother carries the GCK variant, and that consistent with her family history. We were a little bit sad when we tell the result to the patient, because she actually had a lot of euploid embryos. Now half of them she probably don't want to use, but she was actually very positive, and she transferred the first time, and then it becomes pregnancy and live birth, and now she actually called us saying, oh, the baby was born apparently healthy, so we are also happy for her. There are more cases. Probably I'm not gonna spend too much time to go through. We find a microduplication related to seizure. This is another common region for microduplication. We deal with translocation cases. Some patients have a Fragile X risk. They are either intermediate carriers or premature. They want to know what exactly the copy number in their embryos. We were able to run capillary electrophoresis to tell them the exact copy number in each of the embryos, so that they can use more embryos. Than otherwise they were scared that they probably will trash some of the embryos too. So okay, this is the last section. Ethics and future of whole PGT, whole genome sequencing. Definitely, there are advantages. Our assay offers significant advantages by allowing direct screening of embryos, unlike PGTM, which relies on the parental genome to infer the embryo genetics makeup. Our method provides a more comprehensive screening, enabling the detection of de novo variants as well. Our service is compatible with requests for genes outside our standard list, with no additional cost to be incurred for such requests. Also, our assay uses the NGS technology. And as the growing field of genetics, the development of the technology, the database, the study, everything, we think, by nature, our method, our assay will be more and more accurate. Just want to share some cases, some statistics. So among our PGT whole genome sequencing cases, 32% of our patient involves a PETM request. So basically, many patients, they need to do PETM anyway, so they want to better do a broader screen than just one or two locations. Maybe that's how they choose us. And actually, 30% of this patient, 30% of this 32%. So basically, 10% of our total volume are patients rejected by other PET labs due to their PTM requests that cannot be accommodated. And this is actually why we built the company. We can help more people in need, and those people were rejected. Now they can find a place for help. We definitely have limitations. One of the limitations is the database. There are some diseases with unknown genetic causes. Unfortunately, we cannot do screen for those for the patient. If there's unknown genetic cause, then we also cannot do anything for those patients. Another thing is because we're using NGS, so the limitation of NGS apply to us as well. Some diseases are caused by the repeat expansion, right? So besides the Fragile X, we can do Fragile X, but other than Fragile X, we hasn't tried other ones and wasn't able to do it for other diseases. So for those type of situations, the traditional PETM method still need to be applied. For example, collect the parent sample, diagnose report, and also some family members sample with diagnosed report as well. So we need all that so those repeat expansions still need to go through the regular PETM route. So ethics concern, I wrote I think more, but it's actually I can talk about in short. So basically, you can see when I talk about cases, sometimes patient will accidentally receive genetic information of their own. So that's why we require a pre-testing but free genetic counseling and to the patient. We need to talk to the patient, make sure she or he or she understand the risk and the benefit of this assay, especially the risk. So because sometimes the embryo's DNA is made of from both the patient and the partners. So we want to make sure if we see something, this is the risk you will also receive. Um, the second point is some of the findings, positive findings, are manageable or late onset. So for example, the BRCA1, the breast cancer, and also some like diabetes, is manageable conditions. Uh, I hope patient can use us as a prioritization method or guidelines rather than a determinant factor. So basically, if they run out of euploid clean embryos, they can still consider implanting affected embryos as long as they were told about the risk of using those embryos. But as long as they're manageable, I think they need a full consultation with doctors and genomic counselors. But this is what we are thinking. Also, we are worried about the over-reporting as well. So, but so far, we have been very conservative. We do not report VUSs. We do not report also autosomal recessive unless the patient requests. And yeah, I just want to say, people, usually when we talk about whole genome sequencing, the first response is, oh, do you report everything? Do patients still have embryos to implant? So far, yes, we do not report everything, and so far, patient has been, they do have embryos, decent amount of embryo for implantation even after the whole genome sequencing. Uh, for futures, I actually missed the slides for future, but overall, I do think there's a room for PGT whole genome sequencing in the PGT field. You can see we already start taking actions to accommodate patients in need that was rejected by other laboratories. But eventually, I don't think we can replace PGTA. PGTA is still going to be a fast, cheap, and quick screen options. And we screen for deeper things, but sometimes patients are lower risk, so they don't really need us. So this is really for patient who actually cannot find any help, or patient who want to screen more than just counting chromosomes. Yeah, I think that's all my presentation. And thanks for listening. I'm happy to take any questions.
Thank you so much.